Everyone. Thank you so much for joining us again for this great talk by Professor Glen Tibbits. Before starting his presentation and his introduction, I go through some housekeeping notes. As you know, we are recording this talk, and then we are gonna later upload it on our YouTube channel, so you can subscribe the YouTube channel to get the most updated videos and recording from the past talks. Also, there is a question and answer box on the bottom of the screen, so you can go and ask your question there. And then if you want to chat with other folks on this call, you can chat on the chat box. Also, for our next speaker, we have the next Wednesday at the same time, Professor Arash Gradwar from UC Irvine. And he's talking about engineering strategies for structural heart disease. And last but not least, I would like to thank Montreal Transmed Tech for sponsoring this event from almost the beginning of this e-seminar series. And also, if you want to get the most updated information about upcoming talks, you can follow our Twitter account, hashtag TransLBME, and also on LinkedIn. So by that, I'm really glad, and it's our great pleasure to have Professor Tibbits with us today. So after completing his bachelor's degree at McGill University, Professor Glen Tibbits pursued graduate studies at University of California, Los Angeles, UCLA. During these studies, he developed a profound curiosity about electrical and contractile properties of the heart and completed his master's and PhD at UCLA Brain Research Institute. Founded by the American Heart Association, he studied cardiac pharmacology in Niigata, Japan, in year one of his postdoc studies, and cardiac biophysics at the UCLA School of Medicine in year two. He subsequently was appointed as an assistant research professor of pediatric cardiology at UCLA and then assistant professor at the University of Washington in Seattle. When a faculty position opened at Simon Fraser University, he chose to return to Canada after an absence of 15 years. At SFU, he was appointed as full professor in 1992 and a Tier 1 Canada Research Chair in Molecular Cardiac Physiology from 2004 to 2018. The Canada Research Chair position allowed him and an incredible group of trainees to build a research program incorporating the use of human-induced pluripotent stem cells differentiated into cardiomyocytes to understand better inherited cardiac arrhythmias and cardiomyopathies. By that, it's our great honor to have Professor Tibbits with us, and I invite him to share his screen and start his talk. Thank you so much.
How's that? Can that be seen?
Not yet.
Not yet. Okay.
And then if you go to full screen, yeah.
There we go.
Great, thank you so much.
Okay, well, thank you. I guess I can say merci, which is both Farsi and French, I guess. Is that
correct? I think
okay.
Persians and French people understand on this call.
I'm killing two birds with one stone. So merci for the very kind introduction. And as indicated from the title, I'm going to be focusing entirely on using human-induced pluripotent stem cell-derived cardiomyocytes to study hypertrophic cardiomyopathy. This is a very complex disease, and I'll just talk about it briefly. This is one of the most common inherited cardiac diseases we have. It can affect 1 in 500 individuals, and it results in an asymmetric hypertrophy. And what that means is typically when you develop hypertrophy in response to, say, hypertension, some hemodynamic problems, the hypertrophy is symmetrical. All walls, all chambers, whatever the affected chamber is, are kind of uniformly undergoing hypertrophy. This is different. Typically, we see a change in the septum. So as we go from a normal heart here to a hypertrophic heart, you can see the septum is increased even more than the posterior wall here. And this is very common with HCM or hypertrophic cardiomyopathy. And it's independent of hemodynamics. And what that means is hypertrophy is normally triggered by things like hypertension, but these patients could have completely normal blood pressure, but still develop this because of the genetic missense mutation. And it results in myofibrillar disarray. The myofibrils are a component of the heart cells. Fibrosis occurs between heart cells, and it might look something like this. So in panel A, we have a normal heart, and in panels B, C, and D, we have mutations, which are different mutations which cause hypertrophic cardiomyopathy. You can see the disarray, and you can see in panel D the fibrosis shown in yellow between adjacent cells. This is, interestingly, this is the most common cause of sudden cardiac arrest in youth and young athletes. And so, you know, when you see an athlete, a very fit athlete who might have sudden cardiac arrest like we saw in the Euro Cup this summer, the Danish soccer player, more often than not, this is because of HCM, and it could easily be due to a mutation that results in no symptoms until the cardiac arrest. So it can be very dangerous. As I indicated, this is heritable disorder, autosomal dominant, one allele can cause it. And the mutations are typically found in the cardiac contractile proteins, which are shown here. Here we have the thick filaments, largely myosin and titin, or mutations in the thin filament, actin, tropomyosin, or the troponin complex. The general thinking is that whatever the mutation is, it causes an increased affinity of the contractile element for calcium. So here we have calcium. It binds to troponin C to initiate contraction, and that allows cross bridges to form. And so these missense mutations increase the ability of calcium to bind here and put this into a gain of function in terms of the molecular motor. We have some promise of a drug called mavacamten, which is a myosin ATPase inhibitor in terms of clinical treatment. And it's been undergoing phase 3 clinical trials and is definitely showing some promise for certain types of hypertrophic cardiomyopathy. Now, how do we study this? Now, obviously, historically, the mouse has been a very prominent and important player in this, and we can generate transgenic mice, we can knock in mutations, etc. However, the mouse obviously is not a human. The mouse has a heart rate of 600, 700 beats per minute, has very different hemodynamics than the human. And so while there is very useful information that's generated from mice, there is a lot to be learned from other models, in particular those that are human. But dealing with humans per se is very tricky, difficult to get heart samples, and even when we do, they are contaminated by a long history of perhaps drug taking to compensate for the hypertrophic cardiomyopathy. So these aren't ideal models by any stretch. And so the prominence now, excuse me, of human-induced pluripotent stem cells has really been transformative. And this is the work of Yamanaka, in which they took fibroblasts from humans. They cultured the fibroblasts. They infected them with the so-called Yamanaka factors. They became pluripotent within a month to 5 weeks. And then these can be differentiated in a variety of cells, including heart cells, which is what we focus on. And of course, Shinya Yamanaka won the Nobel Prize for this really incredible transformative work in 2012. And that's where we go. we start from here. So iPSC-derived heart cells or cardiomyocytes can be used for many things: drug screening. Colleagues of mine are working on autologous regeneration where they're injecting over 1 billion heart cells into infarcted hearts to try and improve their mechanical and electrical recovery. But we're typically doing disease in the dish modeling and screening drugs that might be more effective for this disease modeling. We've been really fortunate in that we've been able to develop this Cellular and Regenerative Medicine Center here at the Children's Hospital in Vancouver. And there's several aspects to it, one of which is a cardiomyocyte pipeline. Another major component of it is developing pancreatic beta cells for patients with diabetes and insulin disorders. We are able to take blood from patients. We can also have a variety of engineered lines that we use, reprogram those, expand them into human-induced pluripotent stem cells. We can genome edit them to make disease in a dish cells that are really important to study. Now, the genome editing can be to introduce a mutation of interest. In the case of patients that are harboring mutation that we think are causal, excuse me, the genome editing can be used to remove that mutation to generate an isogenic control. And I think most of you are aware of genome editing using CRISPR-Cas9, using a guide RNA. And this has also been another transformative component of doing precision medicine and disease in a dish kind of studies. However, this field is just evolving continuously. It's actually really exciting to see. And so, we started doing genome editing using TALENs. Now most people are at CRISPR-Cas9 by Jennifer Doudna and Charpentier. They also won the Nobel Prize last year for this incredible work. But the work of David Liu and others, Nozomu Yachie, have moved on to other types of CRISPR, which offer certain advantages. We've moved on to prime editing, or CRISPR 3.0, and it has a lot of advantages. One is it doesn't require a double-strand break of your DNA, doesn't require a donor DNA. If you're familiar with the genome editing, you know that there's a PAM site that needs to be in proximity to where you want to cut your DNA, and prime editing is less restrictive in that regard. It's more versatile than base editing, which I'm not going to describe, and it's much more efficient. So CRISPR Cas9 or CRISPR 1.0 requires a double-strand break, and then you rely on the repair mechanisms within the cell to repair that double-strand break. And the repair mechanism that you typically depend on is called homology-directed repair, or HDR, and the efficiency of this is in the order of 1 or 2%. Prime editing, on the other hand, can result in efficiency that exceeds 50%. So it's a lot more efficient and can be, once worked out, can be a lot more effective. So we have genome editing, we have iPSCs. We also have a variety of lines in which we've created reporters that tell us about something about the state of the cell, whether it's genetically encoded reporters telling us about calcium within a certain part of the cell or certain aspects of cellular function. These are really informative. They're really telling us a lot about the disease of interest. And then we differentiate the iPSCs into cardiomyocytes. And this is commonly done by many labs now, and it's a very important technique. Just to give you a little bit of idea, the iPSCs are put through this kind of pathway, and the most common way of doing this is by sequentially activating the Wnt signaling pathway using a small molecule, this molecule right here, this Chiar compound, and then subsequently inhibiting the Wnt signaling pathway and directing the cells to this immature cardiomyocyte stage. And then you may invoke differential protocols to mature the cardiomyocytes. But these immature cardiomyocytes are expressing a lot of the genes that you'd expect to see in a heart: myosin heavy chain, troponin T, troponin I, myosin light chains, et cetera, et cetera. These cells are beating, and they have many characteristics of a normal heart tissue. Just to show you, this work was done maybe 5 or 6 years ago by a PhD student of mine, Sanam Shafatalab, and she just did this incredible work where she had a petri dish here, and this petri dish actually started from her own blood cells. So we isolated PBMCs, type of monocyte, from her blood. She did actually, and then she reprogrammed her own PBMCs into iPSCs, into pluripotent stem cells, and then she differentiated these cells to create her own heart cells, believe it or not. And so this is the pathway I just talked about. She went through this entire pathway in this petri dish of about 35 millimeters. And as you can see, these two indicators of pluripotency drop off immediately as she started differentiating the cells, and markers of cardiac myocytes increased immediately after she started to turn them into heart cells. And within 10 days after the differentiation, you could see the whole petri dish was beating. I remember this day. This was like 6 years ago, and it was just amazing that not only the beating, and you've probably seen online where a lot of people are showing beating, but this beating is really showing an integration of tens of thousands of cells that are in this petri dish. And clearly they're electrically coupled, and we have conduction of the signal across the dish. So not only the cells been differentiated into beating cardiomyocytes, but they're also interconnected as they would normally be in an intact heart. But as I said before, they're still relatively immature. And so the process of going from immature cardiomyocytes to mature is the focus of really intense work for obvious reasons, for disease in a dish, for drug screening, and perhaps even for autologous regeneration. We know there's many aspects that can contribute to this transformation to mature cells. And they can include chemical stimuli. There's a variety of different chemical stimuli. Here is a hormone from the thyroid gland, free fatty acids. There's many ways of using chemical stimuli included right here. Some of them are thought to act through this pathway, the mTOR, by inhibiting it. We can also electrically stimulate these cells, and they will be transformed. We can use nanotopography. We can determine the matrix on which the cells grow to force them to grow down channels. This also has a maturing effect. And we can do mechanical stretching with a certain periodicity, certain amplitude, certain frequency. And then we have a combination of all of these, which contribute in a very complex pathway of going from immature to mature. And just to give you an example of what we do in the lab, this is our maturation protocol. And in the top line are cells that are generated through our differentiation protocol without maturation. And you can see these are the nuclei, and the cells are surrounding them. What you can see is the cells are relatively small. The nucleus is the most prominent part of the cell. Nucleus is almost as big as the cell. And we have very few mitochondria shown here, which is not how a mature heart is. Mature heart is about 35% by volume of mitochondria. This is showing two different maturation protocols. The first one is taurin-1, which is an mTOR inhibitor. And what we can see is an increase in mitochondria shown here, marked by M, and increased sarcomeres and sarcomere alignment shown here in the Z lines are the sarcomeres. We're also seeing other structures we would expect to see in a mature heart. But even more pronounced is a maturation media that we use, which is made up of a variety of compounds, but includes free fatty acids, which is the heart's main target when it's mature, its main substrate when it's mature. And now we can see a huge biogenesis of mitochondria. We see these specialized structures of the sarcomeres shown right here, of the structures which allow the excitation of the heart to be coupled to the contraction of the heart. So this is something that we use. We've actually modified it since we generated these electron micrographs, but it's very promising. So we go through this maturation protocol, and then we can do tissue engineering, which I'm going to only talk about very briefly today. And so the plate that I showed you from Sanam's work showed not only cardiomyocytes that were beating, but they showed this ability to talk to each other through intercalated discs. And within the intercalated discs, there must be gap junctions. And we know that because the transcriptome is expressing the protein. In this case, it's called connexin 43. But also these are functional hemichannels. Hemichannel because one half is in each cell. And we know that the electrical pulse is going from one cell to the next. That's the only way we can describe the conduction that we have, which is pretty high velocity. Now, a lot of the experiments I'm going to show you are using our optical mapping rigs in which we can put dyes or genetically encoded reporters into the heart cells and monitor voltage and calcium in real time in live cells. This was developed by Eric Lin, incredible postdoc in my lab, and it involves fluorescence microscopy using, in this case, the dyes we used were excited by the single wavelength, but emitted two different wavelengths, and he designed this system and he wrote the software to allow us to analyze the electrical and the calcium signaling in the cells. And so this just gives you a rough idea of what the signaling looks like. The black line is the action potential, the period of excitation, and the red line is the calcium transient, which is initiated by the depolarization. And then this is the what part that elicits the contractile process. Now, the real data looks something like this, and these are from 4 different regions of interest in this 35 millimeter Petri dish. So if we look at each region of interest, we can see here's calcium transient. This is the raw data in real time. And this is the action potential shown in an inverse function. So the fluorescence actually decreases. And so through the software, this is reverted. Then if we look at another region of interest, say down here, we can see this. And we can see the differences in time between one region of interest and another, which is reflecting the conduction velocity and potential arrhythmias that occur in one region of interest, but not the other. So this is really sophisticated software that Eric developed and that we use on our heart cells. And from this, he was able to measure conduction velocity from one region of interest to another. And amazingly, under the right conditions, we can get conduction velocities which are about half that of an intact human heart. Surprisingly high. So let's come back to the hypertrophic cardiomyopathy, and we've been studying troponin T variants, which represent one particular aspect of inducing hypertrophic cardiomyopathy. As I mentioned before, HCM causing variants are always sarcomeric proteins. Here is a sarcomere here, and the sarcomere is a component of a heart cell or a cardiomyocyte. These sarcomeres exist within the myofibrils. So here's the heart cell. Here are the myofibrils, which are a subdivision and a contractile specific component where the contractile filaments are housed together into this myofibril. And if we look at the thin filament shown right down here, in higher magnification, we can see the thin filament is made of actin in orange, tropomyosin in dark blue, and the troponin complex shown in these 3 colors right here. Troponin T is shown right here, and this is we're looking at variations in here that cause hypertrophic cardiomyopathy. Now, one of the reasons we study this is not the most common gene, or not the most common target, but it is a relatively common target. But more importantly for us is that this type of hypertrophic cardiomyopathy doesn't show pronounced hypertrophy. The hypertrophy is less than other HCM causing mutants, and it has an increased propensity to become arrhythmogenic. Why is this important? It's important because clinically, one of the ways that clinicians can find out whether your heart is abnormal is by doing an echocardiography. And the echo will tell you whether the heart is hypertrophied. So if at rest your electrical activity is normal and they do an echo on you and an ECG, you may look normal, even though you're harboring this mutation, which may trigger arrhythmogenesis under exercise conditions. So this was of interest to us. This is troponin T, the troponin complex shown right here. So the one mutation that we've started with is called I79N. If you're not familiar, this is on residue 79 of troponin T and isoleucine to asparagine. All of these mutations are heterozygous, so we created these with CRISPR-Cas9, and we found cells which exhibited a heterozygous variant. Now, just to show you phylogenetically, if we go from humans all the way to zebrafish, the isoleucine is conserved over the span, this phylogenetic span. And this actually equates to about 400 million years from the zebrafish all the way through to humans, meaning that through evolution, nature felt that it was not a good idea to change it, or another way of looking at it, changes resulted in serious consequences, and that animals harboring those mutations probably didn't survive as well as those that did not. Now, one way we studied this is using human reconstituted thin filaments, and this was done by a PhD student in my lab, Allison Lee, and she did incredible work. And the reconstituted thin filament, here's the thin filament that I showed you before, and what she did was she used human genes that express each of these, so a human gene for troponin I, troponin C, troponin T, tropomyosin, and actin. She expressed all of those in bacteria. She purified them, and then she reassembled the thin filament in vitro with known components. In this case, the troponin T had the variant of interest, I79N. And what she was looking at were two things. One is the calcium affinity for the complex. And this parameter, KD, is inversely reflecting affinity. So as the number goes down, it means the affinity has increased. So the I79N showed increased calcium affinity, and this was due to the fact that the off rate constant, the rate that calcium comes off of here, is slowed down. And this fact that the off rate constant is slowed down is the reason why the affinity is increased. So this gave us some idea of how the underlying HCM phenotype may be caused. And then Sanam, whose name I mentioned earlier, who developed a lot of this and did incredible work during her PhD, made monolayers of the iPSCs, both wild type, and she's able to stimulate these cells at different frequencies. The wild type, 55, 65 beats per minute, up to 100 beats per minute. You can take them up to 150, 108 beats per minute, and they show this normal functioning, the voltage and the calcium transients. When she made the mutation and she made a monolayer of these cells harboring this mutation, she found something very different, and that is that the cells could not tolerate a frequency of 100 beats per minute. They developed what we call alternans, which is clearly an arrhythmogenic, an abnormal response, clearly indicating that this is a serious mutation. This is just showing you some of the characteristics of the action potential morphology, but one of the major features of it is the action potential is actually shorter at 55 beats per minute. This parameter right here is the time it takes to repolarize to 30% of the normal value. This is 80%. The early phase of the action potential is shortened. We call this triangulation, and that in itself can be arrhythmogenic. Calcium transient was also affected in the I79N. These are both indicative of possible changes which can be causal in terms of the phenotype that we're looking at. Now, the next thing that we did, and this resulted in very surprising results, is we looked at the transcriptome. In other words, how is gene expression affected by this? And For this, we used a type of analysis called a nanostring. Nanostring's a company in Seattle, a spin-off company from University of Washington. And we had a code set made up of 251 transcripts that we had selected. These were genes of interest to us. And so we were able to analyze 251 transcripts in the I79N versus the wild type. This is called a volcano plot. For those of you who are not familiar with the volcano plot, on the x-axis right here, we have the fold increase moving in this direction to the right, or fold decrease in the variant compared to wild type. And then on the wild axis, we have the p-value, or the probability that this is significant. These transcripts right here, shown in red, are increased in a very significant way in the I79N compared to the wild type, and these are downregulated significantly. This is a bunch of genes which were not significantly impacted by just a mutation. Now remember, we're looking at I79N versus its isogenic control. In other words, it's exactly the same, except for one allele carries this mutation. Just to highlight some of these differences, we saw really significant increases in the Notch signaling pathway, which really adds a whole new dimension of how we're looking at this. In addition to this, even though I79N is not thought to create a huge degree of hypertrophy, it is showing a dramatic increase in two markers, early markers of hypertrophy. This is ANP and BNP. These are often used as indices of hypertrophy. Now, in addition to that, we looked at another hypertrophic causing troponin T mutation, R278C. Now, this is a mutation that we could model. So here's the R278C, and we could model this using molecular dynamics simulations. We could not do this at I79N, and the reason being is the molecular structure of troponin T in the vicinity of residue 79 is not known. It's unclear. But we do know the structure around residue 278C. Here is the normal or the wild type structure, interaction between these different components of the troponin complex. Here's the C that's replaced the R, and we can see it's dramatically impacted the interconnectivity of these various components of the troponin complex. It also shows a very phylogenetic conservation of the residue of the arginine at 278C. And what we found is it also shows increased calcium sensitivity and an even more pronounced reduction in the off rate constant. And it shows arrhythmogenicity when stimulated at higher frequencies. And if we plot the action potentials, interestingly, this action potential is at the same beating rate. The wild type looks something like this, which is sort of what you'd expect in a normal heart. But the I79 cells showed a shortened action potential and triangulation, shown right here. But surprisingly, 278C actually showed a prolongation of the action potential. This was not expected, and nor was the transcriptome that's shown here. So this is the I79N, the transcriptome, the volcano plot. I just showed you that. The R278C also had dramatic changes in the transcriptome, and some of the changes were similar to what we saw in I79N. We see an increase in these hypertrophic markers, but not to the same degree. But we also saw a decrease in some of the collagens that were highly expressed, increased in expression in I79N. So in other words, the mutation is triggering differences in gene expression from wild type, but also dramatic differences between these two HCM causing mutants or variants. So just to summarize what I've done today, to date, is we looked at two hypertrophic cardiomyopathy mutations, both of them in troponin T, both of them heterozygous. There are striking differences in these clinically. I79N is thought to be the more malicious of these two. has a higher morbidity and mortality. R278C has a really interesting and very mixed clinical picture, can result in premature death. The patients are often older when they succumb to the arrhythmias, and the penetrance is not as great. However, despite that clinical picture, which is sort of unclear, very clear for I79N, it's a nasty mutation, R278C, not so clear. Despite that, they both show increased myofilament calcium sensitivity, which in the eyes of many is the underpinning of this disease. And they both have a reduced calcium off rate constant. Calcium comes off the troponin complex more slowly, which is the reason why the sensitivity is higher. It's hanging on for dear life. And in fact, the R278C has a higher affinity than does the I79N. However, we have this very different clinical scenario. As you saw, they're both arrhythmogenic, as we studied in monolayers, and they both have increases or changes in the transcriptome, which can reflect these hypertrophic markers, ANP and BNP. But it's unlikely they produce the same phenotype. The differences, as I mentioned, are the action potential morphology and very substantial changes in the collagen as reflected in the transcriptome. Now, collagen is a protein that exists in the extracellular space. It's a very important determinant of the heart's stiffness, and the stiffness of the heart is also a very important physiological parameter. And there's many flavors of collagen. The different collagens that can be formed have different biophysical properties and response to disease. Different collagens can form, resulting in different myocardial stiffnesses. Now, there are certain diseases. One classic disease is called heart failure with preserved ejection fraction, which results from very significant increase in the stiffness. And this can be related in part to the collagen and other factors which contribute to stiffness, which I'm going to talk about in a moment. So that kind of summarized our data. So where are we going from here, our future directions? And when I say our future directions, these are actually where we are right now. I just don't have data that I can talk about. But we're going to be examining the I79N and R278C in multiple genomic backgrounds. So what we did and what most people do is they have a line that's of a specific genome, and they introduce these variants and they compare them between the wild type, same genome, and the one that's harboring the variant. But we know that some people could have this variant and not really have any symptoms. So we are creating these genomic variants in different genomic backgrounds, both in terms of sex, male and female, and also a diversity of genomic backgrounds. How are we doing that? Well, we were very fortunate to have been able to acquire this robocult from StemCell Technologies, and we're the first adopters in the world. So as you may know, StemCell Technologies is the biggest biotech company in Canada, up to $1 billion of sales every year. And they have developed this machine to maintain and passage iPSCs, which is a very time-consuming, labor-intensive, and yet meticulous task that has to be done. And you are a slave to the cells, meaning you have to come in on weekends, et cetera, change media. This robocult completely roboticizes that whole process with the robot in here, incubators, refrigerated baths for the media. And so it can allow us to run multiple lines. In fact, this unit can run up to 100 lines simultaneously with the different variants. And so we can look at the variability with the different genomic backgrounds. And so we're really excited about this. We purchased this, and it should be arriving in our lab within the next couple of weeks. Next thing that we're doing is we're going from the 2D structures, which are these monolayers, in which we get conduction across the monolayer as a sheet, to 3D, more complex bioengineered tissues. And we're doing that with the CellLink Bio X 3D printer, which we bought, and it's supposed to arrive today. And the interesting thing about this is this printer can use the Fresh software. If you're not familiar with Fresh software, this was developed by Adam Feinberg's group in Pittsburgh at the Carnegie Mellon Institute, and they have printed actually intact hearts with vasculature using the FRESH software. And so we're excited to take this on and to look at these variants in a more complex and perhaps a more physiologically relevant scenario. The next thing that we're looking at is the viscoelastic properties of the iPSC cardiomyocytes because of the changes in collagen, but also because we've changed the way the heart cells contract, and that in itself will change the viscoelastic properties. And this has been done by two new PhD students, Yasemin Marif and Cheyenne Janati, and they're working on these. Just to give you an idea, I mentioned the stiffness before, and the intracellular component of stiffness is brought about largely by the titin molecule right here, the titin molecule, which connects the thick filament to the Z line. Extracellular space is conferred largely by the collagen, and we have other factors like the microtubules and the non-muscle myosin. And so the first way in which we're approaching this is to use atomic force microscopy, and this is something that Yasemin is working on, and Cheyenne is going to be modeling the data from here. But these are myofibrils, and the myofibrils are extracted from the iPSC cardiomyocytes. Why is this important? This is important because now we have just the contractile unit, and the sarcomeres have a length dependence in their force of contraction. By using just the myofilament, we can control the sarcomere. We can measure it, and it's very uniform. If we look at the sarcomeres in an intact cell, there's a huge discrepancy, variability in the sarcomere length. But here it can be highly focused. And the myofilament is placed right in here. And the atomic force microscopy comes from the fact that we have a laser beam that is focused on a cantilever, shown right here, a cantilever tip. Here's the myofilament shown right here. It's attached to a glass electrode on one end and the cantilever on the other. And the laser beam comes in and hits the cantilever tip, shown right here. And as the cantilever bends, as the myofilaments exert force, that deflection can be monitored by the return of the laser beam up this path. And this is the unit here. So this is the device. This is called the optical periscope. And if we look at the optical periscope in more detail, so the laser beam comes in here, hits off the cantilever tip. The deflection is reflected in the sound of the light that comes back here. There's a polarizing beam splitter right here and a detector. So we can monitor very small changes in the cantilever deflection. And this constitutes a force resolution in the order of 1 nanonewton of force. Very small. And of course, since we're dealing with a subcellular component, the force is relatively small. So this device has been called by the developer, and the developer is Dilson Razier at McGill University. It's a very elaborate instrument. He calls it the penguin because it looks like a penguin. Here's the beak right here. It's kind of cool. And so Yasemin is working on this right now, taking our iPSCs, harboring these different mutants to look at the biomechanical properties, the viscoelastic properties of these. And the other way we're doing that is by looking at the sarcomere shortening in intact cells. And in these cells right here, there is a GFP inserted into a protein called alpha-actinin that's harbored on the Z lines right here. And so you can see these sarcomeres, and using a program called SarcTrack, we can actually monitor the individual sarcomere shortening. Now, one of the challenging parts of just measuring the shortening is in these relatively immature cells, the direction, the axis of force generation is in many different planes. And so this program looks at each individual small linear component and measures the force and the degree of measures the degree of shortening in each of these and gives you an average for the entire group. It's a very powerful technique, and we've been working with the programmer, Chris Topher at Oxford, on enhancing this. And so this is another exciting direction. And the last thing is to look at the electrical properties And to do this, we're using a Nanoine Patch Liner 8, which is a automated patch clamping rig, which we have in the lab right now, and trying to see why is the action potential different, and how can this be causal in the arrhythmogenicity, and how is it less arrhythmogenic in the R278C versus the I79N. So with these directions that we've undertaken, we're hoping we're going to dig in much deeper and learn not only the mechanisms by which the hearts can be arrhythmogenic, but also the things that would increase the probability, and specific treatments that could be used to try and alleviate these. So that's it for my talk today, and I just want to thank all the people. I mentioned Sanam, who did her PhD in our lab, went to Harvard afterwards to do a postdoc, and she developed a lot of the iPSC work and differentiation, the optical mapping. Allison Lee did the reconstituted thin filaments. Tiffany is working on the prime editing. Homa, Farah Yasemin, and Cheyenne are working on the models of the hypertrophic cardiomyopathy. And we have a great group of collaborators that are really helping us tremendously. So with that, I'll stop sharing my screen and I guess can entertain any questions.
Well, thank you very much, Professor Tibbits, for your excellent talk. I mean, I learned a lot from your talk. There was a variety of technologies that you use in your research that is pretty impressive, and no wonder we see many excellent papers and results coming out of your lab. So I encourage the participants to ask their questions using the Ask a Question box. But before we move on to the question part, I just wanted to remind everyone that our next speaker is Dr. Arash Khradwar from University of California, Irvine, who will be talking about engineering strategies for structural heart disease. So another talk on heart-related diseases. So with that, I'd like to start by asking the questions from the audience. So the first question is from Hoda, and the question that she has is about the application of this model in the clinic. So how would you use the results you obtain from your models in clinics? Maybe if you can just comment on that.
Okay, and that's a great question, and that's the motivation for us doing this. So we're trying to develop the parameters that we think are most important to develop the clinical phenotype, rhythmogenicity, some degree of hypertrophy, et cetera. And then we can go back to our original state, and that is take blood from the patients who are harboring mutations that's causing them to have the disease, and then to look at treatments in a disease in a dish model, and then go back to our colleagues who are treating these patients and find treatments that are specific, in other words, personalized medicine or precision medicine to treat them. But we're trying to develop with these more generic lines a better sense of what are the parameters that are really crucial that can cause the disease. But we will be coming back to very specific patients with the disease and then able to run many patients simultaneously with the robocult. That's our goal.
Okay, that's fair. So I have a follow-up question about this. So these are new humanized models that are being developed and that they're eventually going to be used to reduce the number of animals that are being used. I mean, you mentioned that the animals are not humans there, but they're still, gold standard for in the drug development process. What do you, do you see that these humanized models, or when do you see that these humanized models, in vitro models, will be adopted by pharmaceutical companies? Are there any challenges against using these, or are they accepted by the pharmaceutical companies or the FDA?
Yeah, so That's a great question. And of course, I mean, the advantage of a mouse is it's an intact organism. And if you give a drug, it's metabolized by the liver before it's seen by the heart typically. And we don't have that. And so I think maybe a combination of the techniques. However, you're probably aware, and you probably had some speakers who have done not just organ on a chip, but basically a human on a chip. In which there's going to be liver cells, there's going to be heart cells, et cetera. And the drug will be seen by liver cells first and then by the heart cells. So this is a constantly evolving, incredibly fast developing area that's growing. But there is a group called CEPA, which are developing standards for big pharma to evaluate arrhythmogenic drugs. And it uses iPSCs and other means. As you know, no drug can be approved unless it shows that it doesn't block hERG, one of the important potassium channels in the heart, because there were many drugs, even over-the-counter drugs in the past, which killed patients because it blocked hERG and they went into arrhythmias. So you have to screen, and so like antihistamines, et cetera. So every drug that has to be approved by FDA, you have to show using something like what we're doing here, that it doesn't block hERG. And so that's part of the CEPA protocol. But, you know, right now, about 90% of all developed drugs fail when they go from development and encouraging preclinical studies into phase 1, et cetera. The majority of the drugs fail. And, you know, to develop a drug and bring it to market, we're talking about $1 billion now per drug, maybe 10 billion, which certainly a good deal of this is because the experiments did not allow one to really see what the problems or the efficacy were, in part because it's a non-human model. So there's a tremendous incentive now to go with these models, but I'm not trying to disparage mice experiments. Some of them are really elegant and they are providing useful information. But we are transitioning, and my lab went vegan about 6 years ago. And so, we don't, well, the only animals we kill are the ones the fish we eat. So, but I have a zebrafish model at SFU, which I didn't talk about. So, I mean, that's a good question. It's a good question.
Yeah, because there is still, I mean in the community there's still a debate how to validate these models, and then these multi-organ interactions that you nicely touched on is something that has to be addressed in the field. And then many groups, as you mentioned, are working. Donningberg's group at the Harvard Wyss Institute, and then other groups are working heavily on this. I know that there are groups at University of Toronto, also Radisix groups, and then Gordana's group at Columbia are also working on these multi-organ platforms that are basically that have different organs that interact with each other, and then they're used for affecting the even off-target toxicity of the drugs.
Gordana is doing really elegant work, and in fact, she even has this model where she can show that toxicity can be shown by the heart cells. They release troponin I or T, which is a marker for a heart attack in vivo, clinically used, and she can see if the cardiotoxicity is high. These iPSC-derived cardiomyocytes actually release troponin T into the effluent space. And so it's really cool what she's doing, really elegant.
Yeah. So there's a lot going on in this field, and then a rising star is sitting right in front of us, Human, who is working in this area as well. So I'd like to ask the next question from Ishan. So are the changes caused by the mutations you have studied, for example, the transcriptomic changes seen in collagen deposition, in any way similar or reminiscent of aging cardiac tissue?
Good question. And that's something that we're pursuing. And one thing that I didn't mention is that all these experiments were done on our immature cardiomyocytes. And now we're in the process of maturing these cells and seeing if the transcriptome which we know will change with maturation, but does it differentially change with the presence of the variant? And does that reflect an aged or a pathological heart? The second aspect of that is that we've also grown these cells on a medium with a fixed tensile strength. And the tensile strength of the medium really has a huge impact on the transcriptome. We know that if we increase the tensile strength, so normally the heart has a tensile strength at around 20 or 30 kilopascals, somewhere around there. But you can plate them on matrices, which could be 100 kilopascals, which would represent an incredibly stiff heart, or 10 kilopascals, which is a very compliant. Both of those are pathological conditions. We already know the transcriptome can change just with the Young's modulus of those media. And so we're going to be, Yasin is working on growing these cells with the variants in the face of these different tensile strengths. And we expect the transcriptome to be different as a function of the variant, as a function of the tensile strength. And it's very complex. Is this an aging heart? I don't know, but it's certainly a different heart. Biophysically, viscoelastic, all these things are changing in a way that we don't fully understand yet.
It's incredible. I mean, because it's like the matrix stiffness is very important, and you again you touched on that topic, but along the same direction, there are groups who are working on making these matrices electrically conductive. What are your thoughts on the the electrical conductivity of matrices, and do you think that would have any effect on the cell maturation, or maybe functionality of the cells grown on these matrices?
Are you talking about things like graphene or?
Graphene, nanoparticles, gold nanoparticles, conductive, electroconductive polymers.
I've been watching those experiments, and they're interesting. I mean, graphene to me is just fascinating as a material. It's just incredible, the things that you can do with graphene. The one thing that we were looking at is that you can put certain molecules in the graphene which are optogenetic. You can hit them with a light source, and they will conduct an electrical signal and excite the cells that way. We haven't got into it, and I'm not really sure what the electrical conductivity does to the maturation. Maturation is a very complex phenomenon, and it's the result of many different genes and many aspects of the phenotype changing. So it's a very hard question, but a very reasonable question to ask.
Wonderful. So thank you for answering my questions and others' questions. So we have another question from the from the audience. No, that was the only. So Human, if you have any questions.
Yeah, we have 1 minute left, so I will ask my question very fast. Thank you so much, Professor Tibbits, for this great talk. I learned a lot too. So my question is a little bit general question about the heterogeneity of the differentiated cells after in the culture media. So meaning that we have cardiac fibroblasts at cardiomyocyte, and each time we get different differentiation yield. So does this really make any problem in your work? And also there is a debate also about effect of cardiac fibroblasts on cardiomyocyte metabolic activity and function. Could you please comment on these two questions? One, the heterogeneity of the differentiated culture, and also after that the population, the cardiac fibroblast effect on cardiomyocyte activity.
Wow, you've just opened up Pandora's box. You let all the evil sins out. Now they're crawling around here. Absolutely. So what we do is there's heterogeneity, not only in the fact that fibroblasts and other non-cardiomyocyte, but within the cardiomyocytes, there's still some atrial, some nodal, and ventricular cells. And even with ventricular, that's not just one phenotype. There's flavors within a ventricular. And we know in the intact heart, as you go from the epicardium to the endocardium, the cell properties differ. And so there's a heterogeneity within a normal, a native heart. But what we do is we use metabolic selection, and so that we use high lactate, 5 millimolar lactate, and other things to increase the purity of the cardiomyocytes to about 95%. It doesn't eliminate the issue you talked about, and the differentiation protocol is very hard to control to make it completely the same between. And the other issue, of course, has been addressed in part by Christine Mummery's group, where they actually grew heart cells with fibroblasts and showed that they contributed to maturation, and then they knocked out certain aspects of the fibroblast function and could either enhance or decrease the maturation. So now we're getting into really complex ideas, and I think that paper just opened up the door, but doesn't really tell us everything about it. We can't control the number of fibroblasts other than doing metabolic selection or doing FACS sorting or something like that. And so, yeah, this is a variable that's something we try to assess as best we can, but it's a challenge. And I agree with you. But it makes it fun and makes it exciting.
Yeah, thank you so much. There is a debate on this. So.
Oh, absolutely. Absolutely. Yeah. It's a huge debate.
Huge one on between Zimmerman, Mummery, and other teams. So
yeah, no, I agree.
Yeah. Okay, thank you so much, Mohsen. I pass to you, and then.
Okay, wonderful. Great discussion. I wish we had more time, but Dr. Tibbits is pretty busy, as you can imagine. He has to go back to his group, and then but I really appreciate your time, Dr. Tibbits. I really enjoyed your presentation, excellent results, and then also I would like to thank the participants for joining this event. Just remember that next week we will have Dr. Arash Radvar from University of Irvine as our next speaker. I wish you all have a great day ahead, and then see you next week.
Thank you very much. Merci.
Thank you so much. Merci beaucoup.
Merci beaucoup.
Bye-bye.
Okay. Bye-bye.
Bye.